The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
All right, a little less time today because I gave you a little extra time on that since you were obviously needing a little more time. So for this class and Friday's class, after a little bit on early evolution and behavior, we'll say a little bit about genetics and learning. And then mostly Friday we'll get to the topic of navigation and migration, how animals find their way on foraging trips and other behavior that involves movement along quite a ways from their homes. So there's not much in the readings on these topics, but they come up in studies of the brain and certainly belong in the behavior class. I want to know the most basic multi-purpose movement abilities of animals. We talked about multi-purpose movement abilities already. But I want to know what are the multi-purpose movement abilities that you find even in single-cell organisms. In fact, you find them all the way from protozoa to primates. And they're the ones we're familiar with. Besides elementary life support behaviors and movements within the cell, we have these, even in protozoa, you have locomotor activity for approach and avoidance, foraging and feeding, and for escape from predators. You have orienting movements in response to sensory inputs, and you have various kinds of grasping. Grasping doesn't always mean manual grasping. It can be oral grasping and so forth, and other kinds of grasping. If you're a snake, obviously it's either oral or they have another means of grasping. And then you have various special purpose movements, and these vary a lot more from one between species. So is that any different now for multicellular animals? Actually, all those behaviors listed for the single-cell organisms apply to multicellular animals of all sorts. And I just want to point out that in vertebrates, even if you just eliminate the whole entire forebrain, you find these basic movement abilities are presented. They're represented in the midbrain. They're basic to the organization of the midbrain. Control of locomotion, control of orienting movements, and control of various kinds of grasping movements. But what we've been calling the action-specific potential, that is the motivation behind these movements, the motivations that usually lead to these various multi-purpose movements, then the forebrain is much more involved. So I want to try to summarize basic support systems in very general terms, and then we'll talk about the regulators and controllers of behavior, which are really the motivational states of the forebrain. So I like the way the neuroanatomist here at MIT for many years, Valenauta, described the basic supports underlying all behavior as stabilities. 3 types of stabilities. What do we keep stable? We're talking about internal automatisms and reflexes, what we call the mantle of reflexes. First of all, stability of the internal environment, the vital functions, respiration, breathing. Now, does that involve behavior? Well, certainly, very simple breathing behavior, of course, but if we have trouble breathing, we'll see other kinds of behavior too, try to get enough oxygen. You have circulation of the blood. That's regulated by reflex actions. Regulation of the chemical environment, the blood, the lymphatic fluids, the various extracellular fluids. That involves reflexes, feedback control systems, homeostatic mechanisms generally. And then, of course, we regulate temperature. And that involves reflex actions and involves fixed action patterns of various sorts. And these vary somewhat among across species, but there's some similarity in all the vertebrates. So that's stability of the internal environment. Then we have stability in space, and this includes postural support, various reflex actions that we've talked about. It includes maintaining balance and direction during locomotion as well. That all can be called stability in space, and some people would say it also includes other kinds of spatially oriented behavior, including the orienting behaviors. And finally, stability in time. Now, what if I just am talking about the level of automatisms, including reflexes and feedback mechanisms, then we're talking about, we have to include, though, the temporal organization of the day-night cycle, the circadian rhythms, and their entrainment by the light-dark cycle of the sun. We have longer and shorter rhythms as well, but the major thing is motivational persistence. Without our motivational states, animals in general would flit from one thing to the other, depending on what at the moment was dominant. But motivation tends to persist, and that gives some unity to current behavior. That's all part of stability in time, and it's all regulated pretty automatically. Now, of course, stability in time can be extended a lot when you deal with forebrain mechanisms. I'm asking here for which of the three is the forebrain most important. I've already answered it. Really, it's stability in time here. Okay. So above these basic support systems are the regulators of behavior, the basic controllers we call drives and motivational systems. And I want to just list them. Now, you actually did this somewhat when you talked about ethograms, because it's really a similar thing. If we just talk about the various kinds of what behavioral neuroscientists would call the controllers or regulators of behavior. So we, for example, can start with ingestion drives or motivations, various related drives, foraging, predation, and then a couple kinds of defensive behavior, anti-predator motivation, first of all, and then second, social defensive behaviors, part of social behavior, and then the various drives involved in reproduction. I'm not saying each of these is a single drive, but we can group them. Then we have one that's often left out by ethologists, but not by Lawrence, and it shouldn't be exploration of novelty. This novelty serves the various roles, which we'll talk about in a minute. And then various other motivations, including motivations for elimination, for shelter and nest construction, for sleeping, for play, various social motivations. And we have to add to all these innate behaviors, the learned motivations, because with learning, we also can acquire motivations associated with learned behavior. Something which is not as well understood, but is clearly something we have to deal with. So just adding a little detail, ingestion drives would include both hunger and thirst, the basic ones. And of course, you can get abnormal drives too, but those are abnormalities acquired because of the reward involved and because of the availability of artificial things like drugs. But in fact, animals can also become abnormal in their hunger and thirst. That's why we talk about such things as food addiction, for example. And then the other drives related to feeding are foraging, predatory attack in various predators, prying in starlings is a separate drive. Poking with a spine is separate in cactus finches and so forth. And then for defensive behavior, the various anti-predator behaviors, we have both escape and avoidance, somewhat separate, and we're going to deal with that separately later on. And we'll see a video about escape behavior. And then the various types of defense at the level of social behavior include territorial behavior, maintenance of a position in a hierarchy, achieving a position, maintaining it, improving it, so forth. These are all involve specific social motivations. And then various reproductive drives. Just some essential ones here: pairing, involving roaming or searching, advertising, selecting or responding, which we usually call mate selection. Then mating itself, brood tending, various aspects of parental behavior. Exploration of novelty, just point out some of the roles of that. It plays a role in security and defense. When we see something novel, an animal will interrupt his current behavior. And if he didn't do that, he often would be caught. He improves his ability to detect the source of the predator and the nature of the predator by his orienting movements. And of course, animals, even if there's no predator around, will explore novelty and expand what we call their cognitive map, their map of the environment, their knowledge of the environment, which is crucial for their ability to escape from predators. They're not escaping into an unknown area, but areas they know well, and they know the routes for getting back home or to getting to a hiding place. And then it plays a role in ingestion. We talk about neophobia and neophilia. You know, if animals are starving, they tend to be more neophilic. That is, they will seek novel foods. Whereas normally, if food is relatively plentiful, they will be neophobic. They will avoid novelty. And it does play a role in sexuality as well, more in some species than others. And there's many other motivations that we mentioned some of them before. Okay, now this is another topic about genes and behavior, and I point out here at the beginning that variations in a single gene can result in variations in behavior, and I just want to know what kind of effects on the body does such a gene have to have? What's the very simple answer? If a gene has a specific effect on behavior, like it can be a gene, if the animal has it, like a bee, he engages in cleaning out of the hive, and if he doesn't have it, he doesn't. So you have these two kinds of bees, the hygienic bees and the non-hygienic bees. Well, what has to be different? What does the gene have to cause? The gene by itself knows in single cells, but the single cells aren't controlling behavior. So why is behavior different? How can the gene result in differences in behavior? Very simple. It must be differences in the nervous system, right? Because that's the way we inherit fixed action patterns. Fixed action patterns depend on brain circuitry and other aspects of the brain, including hormones, for example, the chemical environment, and so forth. So that's the kind of effect it has to have. And generally, at least in vertebrates, the effect is on the central nervous system. And I point out, besides hormones here, it could be differences in sensory input and so forth that could result in changes in behavior. That is, if an animal inherited much higher sensory acuity or much lower thresholds to respond to certain types of inputs in the environment, it will change its behavior. Okay, going back to the bees here, the hygienic bees. What are they? Why does a bee colony need hygienic bees? What do those bees do? It's described in Scott, okay, quite clearly. The bees, these are the bees that are opening up the cells, pulling out dead or dying larvae, and getting rid of them, dumping them out. But not all bees do that. At least if you observe, say, a colony of many hundreds of bees, you'll see only a certain percentage of them will actually do that. The others just don't. And Scott uses this as an example of genes and behavior, and he discusses it in terms of two genes. I don't think it's all based on one study that showed that bees, you can create a colony where there's no hygienic bees, and you can have colonies with many more hygienic bees. And the evidence is, from the paper he was studying, that it's based on two genes. Both tend to be dominant. Okay, and if they possess the non-dominant allele, then they will be hygienic. But why are there two genes? Because there's two behaviors involved, and they apparently, according to this study, they inherit separately the ability to do one or the other. They have to, in order to find dead larvae or dying larvae, they have to be able to open the cell in the hive. So he calls that uncapping. And the other involves actually getting into the cell and pulling out the dead larvae. That's another kind of behavior. So two distinct behaviors. And the genetic studies indicate that you can have bees that will uncap but won't clean out. And you can have other bees that won't uncap, but they will clean out dead larvae. So that's why they concluded there have to be two separate genes. Okay, and then he goes on and he points out that in a colony, you can have a colony, let's say, that contains all hygienic bees, but they don't all engage in hygienic behavior. You can have a colony that involves half hygienic bees, but 50% of them don't engage in hygienic behavior. Okay. Furthermore, they engage in the behavior only when they reach a certain age. And the age at which they start their hygienic behavior will actually depend on the proportion of hygienic bees in the colony. So if there's many more, they will engage at an older age. If there's very few, then they will start younger. He spelled out the numbers for that. And I went to one of the studies he was using. The title I listed for you there, Influence of Colony Genotype Composition on the Performance of Hygienic Behavior in the Honeybee. And they described the creation of 4 colonies that had 25% hygienic bees, 50%, 100% hygienic bees, or 0% hygienic bees. Okay, and then this is from their table where they're summarizing many of the results. So here's the percentage of hygienic bees, and the 100N means 100% non-hygienic, so that's 0% hygienic bees. And here they're just listing the number of bees that are hygienic or non-hygienic. The non-hygienic ones are in parentheses, and you'll see that there's 1,600 bees. They adjusted the numbers in these 4 colonies, so there's 1,600 bees in each of the 4 colonies. They went from being all hygienic or no hygienic, and then the in-between ones here, 400 and 1,200 and 800 and 800. Okay, and then here's the statistics on how many of them are actually engaging in hygienic behavior. You can see that if only 25% of them were hygienic, then almost 79% of them engaged in hygienic behavior. If half of them were hygienic, then almost 40%. If all of them were hygienic, only about 19% of them would engage in hygienic behavior in this study. And of course, if none of them were hygienic, then they're only looking at the hygienic behavior of the 2 kinds of bees. But they looked at the non-hygienic behavior, not the non-hygienic bees, non-hygienic behavior in hygienic and non-hygienic bees. And it's very interesting. The non-hygienic bees actually do engage in some hygienic behavior. So that doesn't quite fit the genetic study. So that led me to look a little further, and I found another paper, included one of the same authors, which is how I found it. It was published the same year in the Journal of Comparative Physiology and was entitled Olfactory and Behavioral Response Thresholds to Odors of Diseased Brood Differ Between Hygienic and Non-Hygienic Honeybees. They had a couple different ways of looking at how sensitive they were to odors. They were interested, of course, in their response to the odor of dying or dead larvae. And they found that the hygienic and non-hygienic bees showed different thresholds. The hygienic bees were much more sensitive to the odor. They had inherited different olfactory abilities. But it wasn't that the non-hygienic bees couldn't detect the odor of the dying larvae. They just had much higher thresholds. So it was more difficult for them to detect. So that may explain why the non-hygienic bees sometimes do clean out a cell of dead larvae. They not only can smell, just not as well, okay, but they can also engage in hygienic behavior if they actually detect it. But usually they're not engaging in such behavior because they don't notice it. That's probably much more efficient for a bee colony. You don't need all the bees doing that. What do you need the rest of them for? Well, these are worker bees, and you want them to go out and get the nectar, bring it back to the hive, feed those larvae, because most of them, of course, are alive. unless the colony is suffering from some pathology, which does occur in bees. The study completely neglected the topic that Scott had emphasized, the difference between uncapping and removal behavior. This later study, in spite of one of the authors being the same, they didn't mention that. So I'm not going to make any attempt to reconcile that. This is typical in science. They don't completely satisfy you with any study, but it did explain quite a bit about why some bees engage in the behavior and others don't. All right. A little more about genes and behavior. We talk about genetic fitness. What is genetic fitness? Direct genetic fitness means how many offspring you have. That's one measure of your genetic fitness. But we can also talk about inclusive fitness. How do we define inclusive fitness? It was a major issue for Charles Darwin. This issue, how could sterile workers evolve in social insects? Sterile workers are not having offspring. Sterile workers are the daughters of a queen. Okay? But they share a lot of genes with that queen. In fact, because of the genetics of the Hymenoptera, they usually share 75% the mother. That depends if she only mated with one male anyway. We'll come back to this topic when we deal with sociobiology. It's been a major topic, and it's led to a number of new findings by sociobiologists. But the point is, because the work of those sterile workers, their work for the colony promotes the survival and increase in their genes, which they share with the mother, the queen, they have inclusive fitness. So similarly, if you have a human who has no offspring, it doesn't mean that his inclusive fitness, the broader definition of genetic fitness, isn't present. If he is doing something to promote the survival of his nieces and nephews, for example, of his sister, whatever you see, he's promoting the survival of his own genes. He shares 50% with his siblings. He shares 25% with his nieces and nephews. Okay, that's why the concept of inclusive fitness is so important. It's in fact necessary to explain the evolution of specific types of behavior. All right. Let's say a little bit about learning. It's starting with very simple kinds of learning, habituation, and sensory adaptation. You should know the definitions of habituation and sensory adaptation. The textbook, Scott's textbook, describes briefly just habituation, not sensory adaptation. You can easily find an answer to that, and you should be able to give definitions of habituation. And I'm asking a question there about habituation to different kinds of stimuli. Let's just start with definitions for these two terms. In both cases, there's a decreased response with repeated presentation of the same stimulus. Say I'm touching your back of your hand. I'm sitting next to you. You know me well, and I'm jiggling my hand and I'm touching you. You notice it at the beginning, but pretty soon you habituate to it. Or did you? Maybe you just adapt to it. Maybe it's sensory adaptation. What's the difference? Well, habituation is more central. We can go to physiology and define it that way. It's closer to the motor side. And the level of the response depends on the degree of novelty. So the more the stimulus occurs, the less and less novel it is, the more expected it is, less and less response. In the case of sensory adaptation, and this usually happens right at the receptor level, okay? You get less and less response. The receptors are just not sending as many impulses to the central nervous system. It's a kind of fatigue. That's sensory adaptation, okay? And when the stimulus stops, there's a gradual recovery. The same is true for habituation. There is recovery, okay? But the mechanism is very different. In the case of habituation, it's really difficult to define it as a kind of fatigue, although people have thought of it as fatigue at the more central level, okay? Think of examples of habituation and tell me if you can habituate to both simple and complex stimuli. Can you habituate to several bars of Mozart? You can certainly habituate to a ticking of a clock. You can habituate to cars going by. You live on a busy street like I do. Pretty soon you're ignoring most of it. A particularly loud truck goes by, something changes. You notice it, but most of the time you're not even noticing it. You're habituated to it. You don't normally notice the clock. You don't notice internal sounds in your body unless they change a lot. You habituate to it. What about the bars of Mozart? Can you habituate to it? Yes, you can. You can habituate to complex stimuli too. In fact, in tests of habituation, when something changes that you get a new response. And so in formal tests, you might use series of stimuli. They often use rhythmic stimuli. And they can then suddenly leave out one stimulus in the rhythm. It sounds different. And the animal or the person will make an orienting response. So you can respond to the sudden absence of a stimulus, any detection of novelty. Now, what are the responses? And we won't be able to get much beyond this today. All of these terms are used in describing responses to novel stimuli. The arousal response, the orienting response, orienting reflex is commonly used, especially in the East, and this has been studied a lot in Russia. We talk about curiosity for novelty. That is also a response to novelty. Well, there's responses at the autonomic level, involving the autonomic nervous system. What are those responses? Heart rate change. It's usually a heart rate increase, but if the heart's beating very fast already, it'll actually slow down. Okay? Altered breathing, increased muscle tension, vasodilation in the head, vasoconstriction in the limbs. Interesting that it's different in the head and in the limbs. In the head, where you have all the head senses, it's different than in the limbs. And you can specifically measure that. You can measure the volume of the finger, for example, with a plethysmograph. Easy to hook up a cuff around the finger and measure the volume and record it in a pretty sensitive way. And you will see the vasodilation or vasoconstriction. Then there's behavioral responses, alerting responses, orientation of the head, eyes, and ears, exploratory behaviors. So now we're not just talking about orienting and alerting. We're talking about approach and manipulation, all responses to behavioral responses to novelty. And then finally there's we have a motivation to seek novelty, especially in higher vertebrates. Exploratory behavior occurs caused by a motivation to seek novelty that we call the motivation of curiosity. Okay, so, and I want to point out here that a novel stimulus can be seen as a conflict between expected input and perceived input. And if you look at it, this is the way a cognitive psychologist would look at it, and it corresponds very well to what's happening. And I point out here that the autonomic changes are exactly the same as measured by a lie detector. What is a lie detector measuring? Lies cause a different kind of conflict. In both cases, we're dealing with conflict, and in both cases, we get these same autonomic responses. So if in a lie detection, that's why the skill of the person administering the test is so critical. You know, he can get the autonomic responses just by introducing novelty. And he has to be very aware of that. So we'll start by talking more about other kinds of learning, including associative learning and conditioning next time.